Good morning and good afternoon. We are very pleased to have you join us for this unique Baker webinar that explains historic relationship between 2019 Nobel Prize and Baker Ruskin product line. I will be your moderator today, and I'm joined by Dr. Krista Rantanen and Andrew Skin, who will be our speakers. The 2019 Nobel Prize in Physiology or Medicine was awarded jointly to William J. Kaelin Jr., Sir Peter J. Ratcliffe, and Greg L. Semenza for their discoveries of how cells sense and adapt to oxygen availability. In this webinar, you will learn from Sir Peter Ratcliffe, Krista, and Andrew on their approaches they took to make these discoveries. Welcome, Andrew and Krista.
Thanks, Nevada. Can I have the next slide, please? So I'm Andrew Skin. I'm founder of Ruskin Technology, which is a British company developing and manufacturing products here in Wales, where the big blue arrow is. In 1998, I met Peter Ratcliffe and his team at the Institute of Molecular Medicine at the University of Oxford. And that meeting was a result of chance following direction from Professor Jacques Poussiger and Dr. Darren Richard, who I met in Nice a month earlier. And as you will hear Peter say in his lecture, chance can play a key role in discovery. Ruskin worked with Peter's Oxford team to adapt our technology to deliver the control of oxygen in a controlled physiological cell culture environment, but with precision of oxygen control right down to 0.1%. And that 20 years ago had never been done before. The 1999 Oxford team included names that many of you may know. Patrick Maxwell, who's now Sir Patrick Maxwell at the University of Cambridge, Chris Pugh, who's still a core member of that Oxford team, and David Mole, and of course Panu Jäkelä, from whose lab in Finland Dr. Krista Rantanen came from. By 1999, Ruskin had developed and launched the world's first commercial hypoxia workstation, and today more than 2,000 publications exist that have utilized technology to test disruptive concepts and facilitate discovery. And in doing so, create a new field of science that was the subject of the 2019 Nobel Award for Physiology or Medicine. In 2011, Baker USA acquired Ruskin to create the Baker Ruskin brand we see today. Next slide, please. Here we see the timeline evolution of the technologies that were the core tools for those pioneering this new field across the globe. The picture from 1999 in the top left is the original workstation that Chris Pugh and David Mole worked with, and it's still in operation in Oxford, along with several newer models. Next slide, please. As mentioned, 20 years later, the discoveries of the Ratcliffe laboratory, along with Greg Semenza and Bill Kaelin, were given the highest accolade, the 2019 Nobel Prize for Medicine or Physiology. Now, I'd like to introduce you to Dr. Krista Rantanen. Krista is a disciple of the original Oxford team, and through the work of Panu Jäkelä at the University of Turku in Finland. I've always believed it essential to work close to the science for whom we innovate new tools. And I was proud that Krista was able to join Baker Ruskin as Director of Scientific Applications in 2018 to further develop our close connection to the field. I'll now pass you over to her to introduce Sir Peter Ratcliffe and the story of how the Nobel Prize was won.
Animals need oxygen for the conversion of food into useful energy. The fundamental importance of oxygen has been understood for centuries, but how cells adapt to changes in levels of oxygen has long been unknown. The 2019 Nobel Prize in Physiology or Medicine was awarded jointly to Greg Semenza, William Kaelin, and Sir Peter Ratcliffe for their discoveries on how cells sense and adapt to oxygen availability. In this following lecture, we will hear Sir Peter Ratcliffe explain how they became to make these discoveries. This is a recording from December 2019 from the Nobel lecture at Karolinska Institutet, Stockholm, Sweden. It contains a little bit of history and explains the long journey a scientist goes through in order to make a finding of this magnitude. I will walk you through this story. This is an excellent description, especially for the younger scientists in the audience, of how widely one needs to understand biology and how one then goes on to extracting a phenomena from all this understanding. It also highlights how even the best of them sometimes struggle to get a paper published. In this first section, we will hear about the history of the attempts to understand oxygen and its impact on human physiology. As you will hear, this is not a new concept. Since early 20th century, scientists have done experiments in order to understand the effect of molecular oxygen on cells. So listen to this rather entertaining recap from Sir Peter Ratcliffe on how it all started.
I should like to begin this lecture, since Greg, Bill and I are an Anglo-American trio, with the Anglo-American expedition to Pikes Peak, Colorado, to study altitude, that's hypoxia adaptation, a little over 100 years ago. The expedition was led by John Scott Haldane on the left there, but the heroine for this lecture was surely Mabel Fitzgerald, the only female member of the expedition, not allowed to be a member of Oxford University on account of her gender. But here she is with the men at the summit of Pikes Peak, where there is a very comfortable hotel to base their laboratory. But the decision was that Fitzgerald should work elsewhere to make measurements at intermediate altitude in the surrounding mining towns. The story is that this is because the men were uncomfortable that she was unchaperoned in that comfortable hotel. But off she went on her own to visit those towns that were more or less the Wild West. And these are the measurements she made. On the top there of breathing and below of changes in the blood, in each case showing the very sensitive changes in each of those parameters to small changes in altitude and hence oxygen. This wasn't the first demonstration of adaptation, but I think it was the first demonstration of the extraordinary sensitivity of the hypoxia pathway that we're going to discuss this afternoon. But still they wouldn't behave in growing tissue culture. So great credit should go to Franklin Bunn, who identified in some hepatoma cells, hepatoma from the liver, the other organ makes EPO, that made EPO in tissue culture, and importantly increased it very much in low oxygen. And those were the cells that we, and Greg's alluded to the work in his lab, first used to try to map the control sequences at the erythropoietin gene as a first step in that journey, which we knew must interact with the upstream sensing mechanism. And this was our work on the mouse gene, identifying that 3' enhancer that could confer oxygen regulation on a reporter gene and hence must interact with the upstream apparatus. Chris Pugh and I worked night and day on these extremely cumbersome RNase protection assays that you see here that demonstrated that first step. But then I wanted to take a shortcut. I wanted to be clever and expression clone the upstream bits of the hypoxia signaling pathway by transferring genes from cells that had oxygen sensing, that was Frank's hepatoma cell, to one that didn't. That would be every other cell. And to my surprise and initial irritation, it was necessary to show this cell, the control cell, didn't have the property in the first place. And to my surprise and initial irritation, that was not the case. We found the control cell had the property also. Patrick Maxwell took this work on, and here are some of the protection assays that Patrick did. And they show that every cell, in fact, that we tested ultimately on the right conditions had this property. And we realized that this was of pretty substantial significance, since it implied that extraordinarily sensitive system that had been shown first by Fitzgerald and then by those EPO was present in all cells, which must be doing other things. Since they don't make EPO, they must be regulating other genes in that way. So I was very excited by the work and wrote it up, and not one to take a chance. I drove with the manuscript to the offices of a very famous journal in London, and my aim was to burst in and see an editor, and I believe the editor would immediately see the extraordinary significance of this work, like myself, and agree to publish the work. This is Maunday Thursday, the day before Easter. Agree to publish it immediately after Easter. This is the letter we received about 4 months later on. And for the young people, if they ever say this to you, we had great difficulty finding reviewers. This is good. This means you're in quite a small field. It's going to make life a lot easier for you. Anyway, that was the first bit of my story.
Hypoxia-inducible factor, HIF, was then discovered by Greg Semenza. Like most often is the case, no man's an island, and here too the collaborative efforts were the turning point in elucidating oxygen sensing mechanisms. Let's hear how young Greg and young Peter set out to build the story, and how Bill Kaelin brought the von Hippel-Lindau protein into the equation.
Identification of HIF in those hepatoma cells, and I presented the work that I've just described to you. Here we are at the eagerly contemplating the way forward. Do not be deceived by this photograph. The Lake Lucerne Palace Hotel was the most luxurious place either of us had ever seen before, and we're ever likely to see again until we came here. So anyway, we went back to Oxford, and of course the question was, what were the other genes? And this was our attempt on it. The phosphoglycerate kinase gene, a glycolysis implied in energy metabolism, and there is HIF binding the phosphoglycerate kinase promoter. So glycolysis, of course, is, some people will know, conserved throughout evolution. So we were keen to go back and look in more primitive species. Was HIF there too? And this was another piece of work identifying it in the fly cells, Drosophila, fruit fly, SL2 cells. You can see that rather clear HIF there. We still weren't making much impact with our editorial friends. This paper had the distinction of being first lost and then rejected by an editor of the Journal of Biology Chemistry. But the story in invertebrates is important, so I'll come to later on. As you heard very nicely from Greg, the field exploded and whole new realms of understanding of hypoxia biology following these discoveries, and entirely new fields, and I've set them out just there. But at the time, we wanted to go upstream from HIF, and Greg had importantly identified the encoding genes. So it was possible to ask what aspects of HIF conveyed oxygen sensing, and we, Greg and others, fused bits of the HIF to an artificial transcription factor, what would confer oxygen sensitivity. We found these three regions would all do it. So each of them had to interact with this upstream process and then be transduced from the sensor to a piece of sequence. Now, everyone knew that protein phosphorylation was the means of signal transduction. So there had to be a phosphoacceptor amino acid in each of those regions to receive the signal. But Chris Pugh mutated every phosphoacceptor in this region. It made not a bit of difference at all. This work wasn't all that popular since it was entirely negative, but it did exclude the mainstream hypothesis. So what did those sequences interact with? It turns out there's the ones that interact with the von Hippel-Lindau gene, and well, this was a eureka moment in my lab. In VHL defective cells, HIF is stabilized, and if you put VHL back in, it gains that oxygen-sensitive property. But you know, knowledge builds on knowledge, and this eureka moment didn't happen just out of the blue. Other people contributed importantly to the creation of that knowledge, importantly Bill here. But Greg and I and others were finding these multiple transcripts you've heard about. Bill and others noticed that they were constitutively upregulated in these VHL defective cells from kidney cancer, and they were upregulated irrespective of oxygen. But puzzlingly, they didn't identify the mechanism. We heard about the work in Sheffield on a rainy day. It rains hard in Sheffield town in the north of England. We rushed back to Oxford, obviously to blot for HIF, and this is what we found. Very puzzling, and I presume it was puzzling in Bill's lab too. Another strand of work, Steve McKnight was looking for other pass proteins in the human genome. He found something that might have been a paralog of HIF, called it EPAS, endothelial pass protein. But it needed an antibody to demonstrate whether it's oxygen sensitive or not. Patrick Maxwell in the lab made one, PM9, Patrick Maxwell 9, and Patrick Maxwell 9 was responsible for this blot and the déroulement. Actually, these particular cells, many of them only express HIF2, and that's the demonstration why the blot was initially blank. So this experiment had quite a number of important outputs. Amongst other things, I won't have time for this, but why is the distortion to HIF2 in RCC? As I think Bill will allude to, HIF2 is now a target for RCC, but importantly for this lecture, it was one step further along the oxygen sensing pathway. VHL is a ubiquitin ligase, and in the presence of oxygen, it destroys HIF, and in the absence, HIF builds up. A lot of this work in my lab being Norma Masson and Matthew Cockman following on.
So now they had figured out the HIF-VHL connection, but that didn't quite explain the biochemistry behind this event and what made HIF and VHL bind together in cells. In this section, we will hear how they almost missed to identify the whole reaction because they were conducting the operation in wrong oxygen atmosphere, and how Baker-Ruskin, with its founder Andrew Skinn, made the discovery possible. The story then goes on, telling us how they pinpointed the enzymes responsible for these reactions, and moreover, that these enzymes are very conserved throughout species.
Itself, that was very puzzling to us. Why not oxygen? Andrew Skinn from Baker-Ruskin visited the lab and sold me a hypoxia workstation. I had a bit of spare money from the Wellcome Trust, which is quite important. That's a plug to the scientists to have spare money. And we bought that thing, and Panu and David were then able to reperform the immunoprecipitations with all the buffers equilibrated to hypoxia and perform and then we understood that the reaction was indeed oxygen dependent. So it had these characteristics of oxygen and iron dependent. It wasn't ATP dependent, consistent with the absence of any evidence for protein phosphorylation in the signaling pathway. We were always suspicious of a particular proline residue, but this is one of the moments you live for. We made ultimately a peptide with the proline residue hydroxylated, and you see here with beautiful clarity that you no longer need to treat the extract then, the HIF then with extract binds to VHL very powerfully in any case. This was the work of Yamin Tian. And it had a great significance because that was the evidence that likely the modification was hydroxylation of proline, and that immediately suggested an oxygen sensing mechanism through prolyl hydroxylases, which consume oxygen. And Bill will tell his side of that story. We a little bit more fortunate with the editors these days. We publish those papers back to back in Science. This was the position then, that it was known that the collagen prolyl hydroxylases, oxygen splitting enzymes, they require iron and cobalt, which were those hypoxia mimetics. So we're in some ways rather good candidates anyway, but we couldn't believe the collagen prolyl hydroxylases could do it. Amongst other things, the sequence was wrong, and Johanna Mullaru from Finland was able to exclude that for us biochemically. So what were the enzymes was the next question, and knowledge builds on knowledge, so this is another curious one. We met up with this guy, Chris Schofield, who was to become a very valued long-term collaborator. Now, Chris was working on 2 oxyglutarate oxygenases, but not the collagen enzymes, quite a different thing. He was studying enzymes involved in antibiotic synthesis, and there's one of the first structures. That triad of iron coordinating residues was the signature that enabled him to predict other members of this family, including some that might be prolyl hydroxylases. Meantime, others have been working on mutant C. elegans worms, 145 catalogued mutated worms that couldn't lay eggs. That was the subject of this paper by Bob Horowitz, a previous recipient of this honour. Egg laying, well, a neural, neurosecretory, neuromuscular defect. What could that possibly have to do with what we were looking for? But they catalogued all those mutants. Now, earlier I told you we looked for HIF in flies. John O'Rourke in the lab also looked for it in worms, in C. elegans worms. And this time, critically, he made an antibody. And this is that antibody, rather beautifully showing regulation by oxygen in the worms. Andy Epstein took this project on, and he looked at all sorts of worms. You don't need a license, no expertise, poor worms. They all went in that bell jar, and all of them responded to hypoxia in the way that you see. But one of Chris's mutants, EGL9, look at that, completely constitutive HIF. I can remember Andy Epstein bursting through the office door saying, "Here's your gene." And another of those moments you live for, it sure was. One of Chris's candidates, EGL9, is the prolyl hydroxylase that we're talking about, the oxygen sensor. So that's the system, simply a regulatory oxygen-splitting enzyme that adds an oxygen atom to the proline ring. The alcohol group here then hydrogen bonds to VHL and degrades HIF in the presence of oxygen. That's the switch that regulates all those responses you've heard about from Greg. That's that Fitzgerald first noted in those Wild West mining towns in Colorado. Here she is again, both the acclimatized increased breathing sensitivity to oxygen and the hemoglobin response, both the result of switching the HIF. And we can see here the carotid body that does those breathing responses, expressing HIF protein and lots of HIF mRNA, and those lowly fibroblasts in the kidney expressing HIF and doing the hemoglobin response. That work of other female scientists in the lab, Tammy Bishop, Emma Hodson, and Johanna Lima.
In the final part, we are looking at oxygen physiology and its manipulation for therapeutic use. Also discussed is the extraordinary novel finding that all 4 eukaryotic kingdoms use this system of enzymatic protein oxidation to signal oxygen levels. We also hear how these findings now show promise to become therapeutic modalities.
I'll finish with that at the end, but in the last part of the talk, I want to talk about some new work. HIF, prolyl hydroxylase VHL, in that assembled form, appears only to be present in animals, in metazoan life. But it's since become clear that all 4 eukaryotic kingdoms apparently use this rather odd system of enzymatic protein oxidation coupled to proteostasis to signal oxygen levels, but in somewhat different ways. So here is the system in animal cells I've just described to you. Here is the system in process in dictyostelium, the very nice work of Chris West. It's similar but a little different in the ligase is the target of hydroxylation, not the substrate of the ligase. Here is the system in fish and yeast, the very nice work of Peter Espenschade. Here are different prolyl hydroxylase responding on a different transcription factor involved in the sterol response. But this is one I want to finish with, slightly different in plants. This is a very nice work of Francesco Lacozzi, who showed that plants use a different enzyme, cysteine oxidases, to dioxygenate an N-terminal cysteine in ethylene response factors, a sort of plant equivalent of HIF that enables the plant to make responses to things like flooding. If you overwater and kill your plants, you're probably killing them through hypoxia, and this is the way the plants try to adapt. If you overwater them, they inhibit the destruction of those transcription factors to bring on their own adaptive responses. So the work in our lab on this comes from a chance meeting with Francesco in Rome. Francesco asked me if he could express the animal system in plant cells as an exogenous indicator of hypoxia. I said, why don't we express the plant system in animal cells and see what happens? And this is the very nice work of Norma Masson, Tom Keeley, and Beatrice Guntoli, who came over from Francesco's lab. They fused that plant ethylene response factor sequences to a reporter, and to our surprise, when expressed in human cells, even without the plant oxygen sensing enzyme, it behaved in an oxygen sensitive way. And there you can see induction by hypoxia. The characteristics are different from HIF, but there it was. And when we mutated that target cysteine residue here, we saw, you can see that it doesn't respond, it's constitutive stabilized, as we've seen in some of the HIF system. So the implication was there's a human enzyme, the same as that plant enzyme, doing the same thing. And I hope you'll like this experiment. I love it as a human cell biologist and physician, conducted in Francesco's lab. These are the Arabidopsis seedlings, and this is one with the 4 of the 5 plant cysteine oxidases knocked out. It's looking rather poorly. We identified the human enzyme, and Francesco expressed that in the plants. And there we go, made the plants better, beautiful complementation. So ADO is the human ortholog of the plant oxygen sensor, another type of human oxygen sensor, and this is work in progress. We don't know how important that will be at the moment. We don't know whether there'll be medicine behind it, but it does have some rather interesting targets, the regulator of G protein, and you can see here that in the ADO knockout cell that protein is stabilized, and when we look at G protein signaling in the ADO knockout cell, the regulator, the down regulator, is up, and the G protein signals are down. This is the very nice work of Tom Keeley. So that's coming to the end now. I've and Greg's shown you this amazing human oxygen sensor doing hundreds or thousands of things. I've drawn it in a very simplified way. We've alluded to the plant system, which is also very complicated but works in a different way. It has a reflection in animal cells. And then, interestingly enough, what I didn't show you, HIF it actually also interacts with those RGS proteins. And every time we look at HIF, we find another realm of complexity seems to interact with the acute oxygen sensing system. So out of all that, how could you get the little bit you want to treat a human disease specifically? How can you get what you want? Think about that. But in fact, Bill will go through this, the results of giving those hydroxylase inhibitors in kidney patients are pretty promising. There's newer data from this. So far, these inhibitors show powerful effects on renal anemia with little evidence of general activation against that expectation. You can't always get what you want, but if you try, sometimes you find you get what you need. The words of Sir Michael Jagger and Mr. Keith Richards, of course, by repute, they knew a bit about drug discovery. And what they meant was that there was an element of empiricism, of trial and error in obtaining drugs. Molecular biology, we can take it so far, deliver the targets, but you have to embrace that slight uncertainty. But as Bill will explain, it's looking pretty good at the moment for these drugs. I should thank a lot of people here, past, not quite as many as Greg, I think, but people past and present in my lab. The current group here, very many thanks to them, sunny day in Oxford. The funders, very important. The Wellcome Trust and Oxford University have been with me throughout this time. The Ludwig Cancer Research for the cancer work we now do. Importantly, at the moment, the European Research Council, very valuable message from Britain. Let's stay together. All these things are important.
So thank you, Andrew and Krista, for a very informative session. We would like to thank everybody for participation. And if you have any inquiries or questions, please contact us at sales@bakerraskin.com. You can also visit us on our website, where we have over 2,000 published papers. Finally, you can also follow Krista on Twitter as Dr. Ox at Krista Rantanen on Twitter. Thank you, have a great day, and thanks again for coming.